A very good morning to my panel for today and my fellow coursemates. I am Emiliani Para Anabanyi. Will present my research proposal entitled Antimicrobial Efficacy of Moringa oleifera leaf extracts against bacteria. As an introduction, the research background involved natural remedy as a potential alternative solutions to arising antibiotic resistance associated infections such as skin infections, bronchitis, asthma, and cholera. Moringa oleifera is a widely grown species of monogeneric family of Moringaceae that has a high potential to be turned into a medicine. It has bioactive chemicals that contribute to its medicinal qualities such as anti-cancer, anti-diabetic, tissue protective, anti-inflammatory, antioxidant, and antimicrobial properties. Next, the problem statement of the research include the current antibiotics are less potent to kill or inhibit the antimicrobial resistant bacteria that include pathogens which cause fatal diseases. Moringa oleifera has long been studied for their therapeutic properties and have the potential to be used as natural alternative remedy to eradicate antibiotic resistant bacteria. However, there is limited data regarding the antimicrobial activity of MLE prepared using green extraction method. Therefore, in this study, the antibacterial activity of MLE prepared with different nutrient concentration will be investigated. The questions arise in the research are including which variant of MLE will give the best antimicrobial activity, what is the MIC and MBC of MLE against tested bacteria. Therefore, the objective of the study are to determine the antimicrobial efficiency of MLE using Kirby-Bauer distribution assay and to determine the MIC and MBC of MLE against tested bacterial strains. For the significance of the study, this study will be beneficial in providing initial data on the antimicrobial efficiency of MLE prepared using Bacillus subtilis biofermentation method. It will enhance data that contribute to the application of MLE as natural remedy to eradicate antibiotic-resistant bacteria. Moringa oleifera is an indigenous species of the Indian subcontinent which belongs to the family Moringaceae from the genus Moringa. It is widely grown in the foothill of Himalaya mountain, which is now extend to all over tropical region in Southern America, Africa, and Asia. Each part of the moringa plant, including leaf, flower, bark, seeds, and root, is effective as a medicine. It is also called as miracle tree as it is believed to be a great supplement due to its high protein value. Moving on to the properties and mode of action of Moringa oleifera. Moringa oleifera is a bacteriostatic agent as it has bioactive compounds such as flavonoid, alkaloids, triterpenes, polyphenols, terpenoids, and tannins that are able to inhibit bacterial activity. Flavonoid has been reported to denature protein and damage the bacterial cell wall, which result in the repression of bacterial growth. Tannin can also attack on the polypeptide of the bacterial cell wall, which result in the lysis of cell due to osmotic and physical pressure, while terpenoid will act by reducing the permeability of the bacterial cell wall by forming polymeric bond and damaging the porine on the outer membrane of the bacterial cell wall. The mode of action of an antibacterial agent is including inhibition of bacterial protein biosynthesis, cell wall biosynthesis, nucleic acid synthesis, and destruction of bacterial membrane. In addition, an antibiotic agent can attribute one type of mechanism or mix of different types together. The next part of the literature review is about antibiotic resistance. According to Centers for Disease Control and Prevention, antibiotic resistance takes place when microbes overcome the antibiotic designed to kill the microbe. There are two courses of action for bacteria to resist the effect of antibiotic. First, bacteria interrupt the action of antibiotic from reaching its target via efflux pumps, enzyme, and by decreasing the permeability of bacterial cell membrane. Secondly, bacteria alter the target site of the antibiotic by camouflaging, expressing alternative protein, and generate different form of structure the bacteria need. Now, we are in the last part of the literature review. Green extraction method is the discovery, design, and application of chemical products and processes that minimize or eliminate the use and production of harmful compounds. It is simple, safe, low cost, repeatable, adaptable to a variety of applications, and assure a high quality of product or extract. Our provider Tuah Lab use microbial fermentation with Bacillus subtilis, which is one of the green extraction, to prepare MLE. The fermentation convert complex chemical compounds into simpler ones such as amino acid and peptides. Lastly, Bacillus subtilis help in increasing the extraction of the phenolic compounds of Moringa oleifera. For research methodology, we will start with the preparation of media such as nutrient broth, nutrient agar, Müller-Hinter broth, and Müller-Hinter agar. Next is bacterial revival and isolation of the tested bacteria from glycerol stocks on nutrient agar. The bacteria that will be tested is Bacillus subtilis, E. coli, Pseudomonas aeruginosa, and Staphylococcus aureus. The method will continue with the preparation of MLE on this diffusion assay with variable concentration of MLE ranging from 15.63 mg per ml to 500 mg per ml. Ciprofloxacin will be used as positive control and distilled water as negative control on every plate. And then this diffusion assay will be conducted. The 4 bacterial strains that are successfully inhibited in this diffusion assay will be used for MIC and MBC determination. Broth microdilation method will be used to prepare 96 well microtractor plate with MHB for determination of MIC. Next, MBC will be determined by subculturing or pipetting 10 microliters of each well from MIC into MHA. And finally, statistical analysis using ANOVA as all tests will be run in triplicates. End of my presentation. Thank you for listening.